So hi, I'm Nathaniel Butterworth from the Sydney Informatics Hub, and there's a couple others from the Informatics Hub too who'll be helping out online, and also Jake Chen from the. I'll let Jake introduce himself shortly, but I thought I'd just go through a little bit of housekeeping. We sent out some setup instructions. Hopefully, you've managed to do that, but if not, that's okay. We'll kind of step through it and talk about what those instructions actually meant that you did. And the first half of the course will be not as technical, I guess. So the second half of the course will require a little bit more Unix and technical skills. So that's where we'll sort of slow it down and go through what was required for those setup instructions. So if you haven't done them yet, that's totally fine. Yeah, so feel free to ask questions as we go along, interrupt and everything. We've got 3 hours for the session today, so that should be plenty of time. We'll probably do an hour, have a 10-minute break, another hour, and then a 10-minute break, and then the final hour-ish. So we'll try and aim for that, so you can plan your mornings accordingly. Please, respect everyone here. It's a totally open environment, though. Ask stupid questions. That's what we're here for learning and sharing knowledge. So look forward to anything like that. And yeah, we've got a bit of an intro looking at AlphaFold on Google Colab, followed by Artemis and HPC specific examples. There's some additional topics here which are kind of we'll talk through the notes, but we won't go into them too deeply. But if that's sort of more your interest, they are there, and we can also ask questions. So we should have plenty of time for questions as we go along. Yeah, so just a bit more on the Sydney Informatics Hub. We're a core research facility at the university. So we take care of things that engage researchers to help them better their research, I suppose. So we are kind of like internal data science consultants. We're bioinformaticians, we're statisticians. We work on the high performance computer a lot, and so we will collaborate with researchers and research groups to help them do informatics style research. So you can engage with us in a bunch of different ways, and the trainings that we offer is just one way that we do that. And I'm calling in from Gadigal land at the moment, so I just like to pay my respects to elders past and present, and just acknowledge the continual search for knowledge and understanding that has been going on for generations around Sydney and the various campuses that we're probably all calling in from. So with that, I might pass it over to Jake, who's going to sort of kick things off. So Jake, please feel free to introduce yourself, and we'll go from there.
All right, thank you very much, Nathaniel. Yeah, so like Nathaniel said, my name is Jake. So I am submitting my PhD thesis in about an hour's time. Yeah, so I'm currently, I guess, still currently a PhD student, but I am completing that now. And so my research for the past 3 years has been a lot on modeling, supercomputing, of studying ion channel receptors. So specifically, so proteins that are present in the cell membrane, and then they allow the flow of things like ions and whatnot. So specifically ligand-gated ion channel. And so I've been one of the first users essentially of AlphaFold at the university. And yeah, so we managed to, so we had a problem where we wanted to model some proteins, but it was just impossible using traditional methods like homology modeling. And so, yeah, so basically we turned to AlphaFold 2, and that's sort of, and then we went down a rabbit hole from there. And now, yeah, so we've got quite a few papers, and I've also presented this year in the biophysics conference in America to a sold out audience of 6,000 people. So that was quite an experience. Yeah, so Nathaniel just invited me to give a short introduction into AlphaFold and sort of the theory behind it. So I've prepared some lecture slides from the content that I give to my first year students. And yeah, So before I start today, like Nathaniel did, I'd like to just acknowledge the traditional owners and custodians of the land, the Gadigal people of the Eora Nation, and like the acknowledgement in my thesis. So the supercomputer that I work on, NCI Gardi, so Gardi actually means to search for in the language of the Ngunnawal people of Canberra. And so that's essentially what the supercomputing and the work that SIH has done, what that enables us to search for things, both at the macro scale, climate change, and also at the micro scale. So things that you'll see today with AlphaFold and looking at proteins and atoms. Okay, so I will just bring up my slides really quickly. So I'll just share like this. I'm on my laptop right now, so it's a little bit difficult to get to the full screen view. But yeah, anyways, so yeah, so AlphaFold 2, it's a AI-based method. So all of you here would have heard about something about AI. So Chat GPT, that's the main thing that everyone's heard of nowadays. But also other things like using AI algorithms, self-driving cars, Teslas, things like that. But I think a lot of people don't actually know what AI is and what AI can do. So this is just a few slides to sort of introduce you to that. So artificial intelligence, so protein structures, they can take years to determine. So for those of you who are not in the structural biology field, proteins, they follow certain fundamental rules in nature. So how atoms, the alpha carbon, how nitrogen, oxygen, how these things are oriented, they have certain ways of orienting with each other. So things like angles, the torsion of the bonds, and whatnot. So there are certain fundamental principles in nature that is always followed in terms of energy. And so we can predict these trends in nature using big data and deep learning. And so that's where AlphaFold comes in. So the first revision of AlphaFold is based on an attention-based model. So an attention-based model is essentially a development of AI architecture. So I won't go too much into the more technical details today, but if you're interested, you can look that up in your own time on Wikipedia or whatnot. So it was essentially one of the first generation attention-based models developed by DeepMind, which is sort of like the AI research of Alphabet, which is Google. And so the initial AlphaFold is able to predict the protein structures fairly well. However, it was not very accurate. And at the CASP 13 competition that DeepMind entered it into, it did not garner a lot of attention at the time because it performed similarly to previous methods that I mentioned previously, like homology modeling, where you take a template and then you sort of mash a new sort of sequence into that, and then using that to sort of then build up a model. And so the scientific community at the time sort of just dismissed it as, oh, it's just another fad. It's not really, a useful tool. And so DeepMind took that and then went back to refine on their initial architecture. And then they released a new generation of attention-based model. And that was released as AlphaFold 2. And so they entered the CASP 14 competition and they essentially swept the floor for every single category that was present. So things like the single protein predictions, multi-protein predictions, protein-protein binding computations. So all of those, they essentially won every single prize, first place and by a wide margin as well. And that was back in 2020, late 2019, early 2020. So that was when people sort of paid attention to this and say, okay, this is, a real competitor to, what we're doing currently. So yeah, so here's just a very, very dumbed down schematic, I guess, of sort of what we're doing when we put in a command into AlphaFold 2. So here we have a uniprot sequence. And so, yeah, so you can see that it's just a sequence of letters. And so this is the primary structure of the protein. It's just a bunch of letters, essentially, of the 20 different amino acids arranged, in order for this protein. So this protein is one that I'm actually working on for one of my projects. And I'll show you the structure that we obtained later on. And then this arrow symbolizes the black box of AI. So something happens in the background. If you're not a technical user, probably don't care. You probably just care about the output. But the arrow symbolizes the black box. So something happens in the back, and then it spits out a 3D structure of a protein. And so this one is actually taken from the UniProt database. So UniProt's established a predicted protein structure database where they've ran AlphaFold on every single protein sequence in the UniProt. It has its goods and bads. So it's good because you can sort of see what it looks like, but it's bad because it has a lot of limitations, which I'll go into a bit later. So opening up that black box, this is what AlphaFold looks like. It's a little bit scary, but I just want you to focus on the main groupings that are present. So that is the embedding group. So this group here, embedding, trunk, heads, and the final structure. So there is 3 key steps in AlphaFold. So if you take nothing else away from today, that's the 3 main steps. And understanding how AlphaFold works is also going to help you a lot in your research as well, because simply because you can start making changes to the architecture later on. Say, if you're, looking to work more advanced in terms of this field, yeah, so that's a possibility for you as well. You can actually modify the underlying architecture, some of which I'll mention later. But so this is just a general sort of overview. So first, the embedding module. So what the embedding module does, it performs essentially a very fancy multi-sequence alignment. So where it looks up all, so numerous databases. So databases such as UniProt 90. There is a custom curated database called the Big Effing Database, BFD. And then there's also the protein data bank as well, where it takes all protein structures from the protein data bank in terms of coordinates and in terms of protein sequence. So that amounts to just over, as last time I checked, just over 3 billion entries, so protein sequences from all species, marine databases, and also soil databases as well. So these are the 3 main ones that might be of interest to, you might be familiar with, but there are other databases that is including as well. So over 3 billion entries, and it's constantly growing as we're depositing more data into these databases. And then what it does is that it will take your input sequence. So say, for example, I would like to put in a test sequence, and then it will then develop a multi-sequence alignment or an MSA, which will tell us what sequences are of interest and what is important for the prediction of our protein sequence in particular. So we'll highlight regions of the protein for the next module, which is the attention-based training module. So we'll highlight which regions are important for our protein, and then it will pay specific attention to those regions in particular. Next is the trunk module. So what this module does is that it processes and then trains the neural network on the bond angles, the residue angles, and other characteristics that's important to our protein structure. So that is where the PDB structures are of interest, because they have those 3D coordinates available. And also it looks at, so that is then fed into the main training module of AlphaFold, which is what they've called EvoFormer. So the EvoFormer module, it looks at the data that's generated and determines that what parts of the protein are important. So it may be a transmembrane domain, it may be, say, the ligand binding domain, it may be whatever. And then this is recycled over and over to improve the result output. So AlphaFold, it's, like all AI methods, so it's constantly teaching itself. So it's constantly repeating the same task over and over again in order to find trends in the data. And this is especially true for AlphaFold. Then what it does, so unlike homology modeling, where you have a template and then you sort of mash that, your new sequence into the previous template, and then introduce things like bias and whatnot. So the structural module of AlphaFold is slightly different from the previous methods, where the amino acids in space, they're treated almost like a gas. So they don't have any sort of there is no like context as to where things are. Essentially, all they care about is the orientation of the residues in 3D space based on the sequence information and based on what the architecture, the previous Evo formal block, has told the module to generate. And then once all of those residues are placed into 3D space, so now you have a whole bunch of amino acids just floating in 3D space, then the bonds are connected. So the backbone is connected, and then it forms the protein. So this is great, but a big criticism of early releases of AlphaFold 2 is that because it has no context of where things next to it are, so the amino acids are just placed in 3D space. So a lot of times atoms are inside of each other, right? And so that's one of the major problems of earlier releases of AlphaFold 2. And that is where the new modules, which is called minimization module, comes in. So it's implemented in the release 1.1 of AlphaFold 2. So what essentially it does, it runs a cycle of a very short MD using the AMBER protocol. And what it does is that it generates a relaxed structure of the protein where things are now no longer inside of each other. And so this creates a protein which is a lot more, useful and workable as a, end user, research end user, for AlphaFold end user, whether you're a structural biologist, whether you just want to do, drug discovery design, or whatever you want to do with it. It's a lot more useful information compared to atoms inside of each other and like aromatic rings like interconnecting with each other. Okay, so earlier I mentioned some implementations. So so NCI Gaudi has a implementation of CollabFold on their server. So you have some Gaudi time that you would like to burn, you can run on that. So I've got quite a lot of experience running that. And also on Artemis as well, we have a local system. So that Nathaniel will go through a bit later on. Yep. And then also there's the Australian Biocommons one as well, which is a little bit more limited because as far as I'm aware, Nathaniel, I think they still on the single protein predictions.
Yeah, that's right.
Yeah. So if you want to use multimeric predictions, then the main one will be Artemis and Gaudi if you want to use a local implementation. But if you're just looking at a single protein, you can use the Australian Biocommons implementation as well. And then there are some online servers, so things like DI-TASA, CollabFold, PEZI folding, and AlphaDoc. So DI-TASA and PEZI folding, these are improved iterations of AlphaFold where they have, on top of the 4 modules I mentioned to you earlier, they have a pre-processing and a post-processing module as well, which improves the accuracy dramatically. And CollabFold, which I'll walk you through just a bit, is built on Google Colab, and so it's a lot more accessible compared to these HPC implementations. And then AlphaDoc is a sort of a wild card here because it uses AlphaFold to predict protein-protein docking, which is a new application that we're currently using as well. So for these third-party implementations, the main downside is that you are running this data on someone else's computer. So if you have some very confidential research data that you're on a patent or whatnot, that might be a consideration for you. But if you would like to just test that out, these are other platforms which you may wish to consider as well. So what can AlphaFold be used for? So some proteins are just physically impossible to characterize in vitro. So using cryo-EM, using crystallography, some of these proteins are just physically impossible. They're impossible to characterize, or they just fall out of solution, or they just aggregate and nothing will ever keep them happy. And so they're impossible to study structurally. And so using AlphaFold, you can at least visualize these proteins and then use other methods to validate your AlphaFold structures. Fitting of cryo-EM and X-ray crystallography maps. So when I was in America earlier this year, that was a big thing that a lot of the American groups are using, where they use AlphaFold to speed up their workflow for cryo-EM and X-ray crystallography work. And some groups have told me that previously they will take them, months to try and fit it, fit a protein into a cryo-EM map, whereas nowadays, with AlphaFold, it takes them, like less than a week to get the same results, if not better, compared to what they would have done manually. So that's a groundbreaking for those, if you're interested in those sort of research. Like I mentioned, protein docking as well. So AlphaFold is somehow able to predict the sort of protein-protein interfaces. We don't quite know why, but it's just able to predict these really well, even better than dedicated protein-protein docking software as well. And so, yeah, so we're currently exploring the use of that as well. Sampling of protein loop conformations. So these are regions of very flexible, areas within the protein. And so using AlphaFold, you can actually generate multiple conformations of these. And typically it would find the lowest energy conformation. And so, yeah, so if you would like to do some sampling of these loops, that's another method you may wish to look at as well. Determination of protein flexibility. So AlphaFold can generate metrics which tells you what areas of the protein it thinks is flexible and what it thinks is not. And so, yeah, so that's another really useful thing as well. And also artificial proteins as well. So things like artificial linkers and whatnot, which I'll show you in a bit, some of the work that I've done. Yeah, and so that's another possible implementation as well. So is AI the future? So I just put in a like cartoon, XKCD. So we like to think of AI as sort of this, omnipotent, thing where it's out to get us essentially. But that's not quite the case. There are limitations of AI. So like this comic here said, like basically you're just pour all the data into a big pile of algebra and maths and then get the answers on the other side. And if the answers are wrong, you just change things up a bit and then until it looks right. So yeah, so that's basically AI. And as the founder, like the founder of OpenAI, Ilya Sutskever, so he is the CEO of the company that's developed ChatGPT. So like the tweet that he's posted, machine learning is just statistics on steroids. Lots and lots of steroids. So we need to keep that in mind. Machine learning, it's essentially running a lot of fancy maths on a lot of data and then using that data to train things. And so we need to keep that in mind while we're looking at these data to make sure that we're not getting too ahead of ourselves. And so on that, what's some of the limitations of AlphaFold? Well, AlphaFold can't be used to predict ligand binding. So you can't put in a ligand and then just say, oh, can you predict this? It also can't predict apostate proteins. So, well, it can't predict ligand binding poses. So what does a binding site look like if there's a ligand inside? What does a binding site look like if, it's not going to ligand inside? So it's basically, it will always predict the lowest energy structure, which may or may not be applicable to you. It struggles to predict open and closed state ion channels. So that's something that we sort of discovered the hard way. And so there are workarounds to this. So the GPCR people, because they've got the luxury of having a massive database of GPCR structures, they're able to essentially trick AlphaFold into training only on that database instead of on the entire PDB. And they're able to trick AlphaFold into generating these open and closed states. However, then you're generating, like potential bias. Like, are we seeing what we're really, what's the real structure, or are we just, pushing it to generate something, right? Something that matches what we think we should be seeing. And so, yeah, so workarounds are modifying dropout, layer modification. So you'll have to go into the architecture itself to play around with that. Changing the number of recycles, changing the databases like the GPCR people did. But there's no easy workaround. So you can't generate open and closed states and compare those. So it will always generate the lowest energy state, which for most things is the closed state. AlphaFold can't predict RNA or DNA or their binding to proteins. So if you're looking at a, say, DNA polymerase, which binds to DNA, it won't be able to predict the correct structure when it's bound to, say, DNA. because it doesn't know what a DNA is, right? So because it's only trained on amino acids, so it can't predict any of these things, which given the current interest in RNA research may rule this tool out for you. And also the big consideration is that resources in terms of running AlphaFold. So CoLabFold can't run on large proteins, and so therefore supercomputers are needed. So CoLabFold is the web service that I mentioned. So the maximum I think is currently around 1,400 to 1,500 residues, and so we will need to use a supercomputer. And then it's sort of, do you have the resources to run that? So you can run on Artemis, you can run on Gaudi, but then Artemis has the fair use weighting, and Gaudi you would have to apply for grants and NC Mass and adaptive schemes and whatnot to get onto Gaudi. So if you don't have that, you would have to buy your own resources or AWS or Azure, as Nathaniel will go into a little bit. And so sort of, do you have the resources to run it? Because these things, they're not, something has to be computing in order for these AI things to run. And so, for an example, ChatGPT burns through almost, I think, around just under $10 million a week in terms of computing costs. So that's only one service of one company. So if you think about that and you think about all the other AI tools out there, it's sort of, is that feasible for you and your research? Okay, that sort of comes to the end of the presentation. I think I'll just stop to see if there's any questions from anyone before I move on to the next part.
Yeah, I've got a quick question, Jake. Thank you for that. How does AlphaFold work on short proteins, like less than 100 amino acids?
As in like how fast it goes, or as in like how does it predict those?
Like how accurate is it for small proteins?
It is really accurate. So sort of when AlphaFold falls apart is when it goes over 4,000 amino acids long, but below that it's got no problems. Yeah. So I've seen people labs using it to predict like peptides when I was in America. So and then yeah, using peptides because that's a big field of research right now. So yeah, so peptides down to 20 residues long, all the way down to massive protein complexes of 3,000, 4,000 residues. Yeah.
Yeah, so there's no increased false positive rate with short proteins.
No. So somehow we don't know why it does it, but it's able to just predict the low. It understands energy, so it's able to predict the lowest energy conformation. So that may or may not be what you're looking for. Yeah.
Thank you.
Yeah. All right. There's no other questions. Daniel, are you happy for me to move on to the next part, the CoLabFold?
Sounds fantastic. Yep.
Sounds good. All right. The right window. Yep, there you go. Okay, so this is what CoLabFold looks like. So for those of you that just want to, sort of play around with it, you're not really sort of sure if you're sort of committed to this, you're wanting to jump down the rabbit hole and spend a whole year working on this. So this is a quick web-based interface, and it's a lot easier compared to HPC implementations because it is a graphic interface. So you basically press buttons and let things happen pretty much. So yeah, so what you do, so for those of you who are not sure how to get here, so basically if you just Google AlphaFold 2 CoLabFold, and it will bring you to this web page. But I think Nathaniel, you've also got a link to this in the materials.
Yep, and I just posted in the chat as well.
Yep. Cool. Okay, so how CoLabFold works is basically a Jupyter notebook. So for those of you who are not familiar, you basically just press these buttons on the side and then it will just run it pretty much. So yeah, so in this case here, let's just take this query sequence and just shorten it down to maybe say like, I think that's around 15 residues, 10, 15 residues. And I'll just do one cycle of relaxation. So yeah, so this is where you would put in your sequence. So for this example here, I'll just put in a really short one. Feel free to put in whatever protein you're interested in doing. And then you'll just have to put in a colon to separate it if you want to model a complex. So if you would like to do a multimeric prediction, feel free to do that. Yeah. Job name, that's fairly self-explanatory, and number of relaxers. So that's how many times, so like I mentioned earlier, the final module, the minimization module. So how many times do you want to run that? So we'll just run it for once, and then, yeah. And then template mode, so you can use templates. You can also detect templates in the PDB, or you can use your own template. So if you would like to influence how it's predicting this structure, feel free to do this. But if you would like to just let it do its own thing and see, what it wants to do, then you can set that to none. So if you'd like to see what it's actually doing, here's what's actually running. But for the purposes of today, I'll just hit run. I will just run anyways. So yeah, so AlphaFold, yeah, so there are some pros and cons. So the pros I've mentioned earlier, it's very, very easy to use. It's very new user friendly, and it's also very accessible as well. So basically anyone can come in and just run jobs. The downside is that Google Colab is an interactive environment. So if you're predicting a particularly large protein, you can't simply just plug something in and then just go and do something else for the day and come back. So every 5 minutes it will prompt you to say, hey, are you still there, sort of thing. And also the main thing limiting AlphaFold's usefulness, if you don't want to pay for it, is that the GPUs are capped at a certain time limit, and also the GPUs are quite old. So you can only predict a certain size. So up to I think a couple hundred for the free plan, and you can go up to 1,000 or slightly more for a paid plan. So that's something that you may wish to. You may think you may wish that if you have the funding, you may think that that's a good trade-off. Yep. Okay. So when it's done, it'll have a little green tick next to it, and that's when we know it's complete. So next, we will install the dependencies. So this is where it installs all of the code. And so, yeah, so this may take a while. So I think what I will do is I'll just walk you through the workflow for today, and then you can explore at your own time, or during the workshop. If you're really lost in the second half, you can come back to this and play around with this. So after that is done, so say if that is complete already, it will generate the MSA options. So how do you want to do the multi-sequence alignment? The default I find works quite well. So MM6, so that's a algorithm to search the database. Pairing mode, usually I will just stick with unpaired and paired. So pair sequences from the same species and also unpaired. But usually the default parameters for this works pretty well. Okay, next one is the actual, so once you finish installing that, then you go into the part where you actually run, put in the settings for your run. So model type, so auto, it will automatically detect what you're trying to predict. So if you just want to predict a single chain, like what I am doing here, so I'm just a very short protein sequence, you can just use PTM. Otherwise, I would use Multima v3 for a multi. So if you want to do a protein docking, or when there's 2 or more protein sequences. Number of recycles. So that comes back to the Evo former block that I mentioned, where the number of times that it's being fed back into the Evo former block, where the output is being fed back into the input. And the more you do it, the more accurate it is. Typically, you will need to benchmark this by yourself. So for the protein that we work with, so we spent about a couple of days benchmarking this, and we found that for our protein, 25 cycles was the ideal value. But for your protein, you might need only 3. You might need, say, 1. You might need, 50 if you're working with a particularly, weird, wacky protein. So that's really, you'll need to play around with that. So it's not a plug and play solution where, yeah. So there is a little bit of, like, you do need to engage with it a little bit, and play around with the settings a little. Yeah. Okay, so recycle early stop. So this is a rather new setting where if it reaches a certain threshold of accuracy, it will automatically stop it. Yeah, so auto is usually okay. Once it reaches 20, 20 is sort of like the cutoff point that we've identified, but your protein again may be different. So you may wish to set this to something else. So like I said, your mileage may vary. Peering strategy, greedy, works quite well. So it will peer any taxonomical subsets if you just want to include everything, pretty much. And then the sample settings, the MSA, so I would recommend leaving that to auto. And then the number of seeds is how many times you want to randomly, how many times you want to generate things. So each different seed will have a slightly different value. And so it will generate something that will look slightly different. So you may find that you want that if you want to sample different conformations or whatnot. Using dropout may improve results. If you find that using all of these settings just doesn't cut it, you may consider using dropout. So what that will do is that it will, during the Evo format, the recycling process, it will essentially eliminate data, which it thinks is quite poor. So most of the time you may not need to use it, but sometimes, yeah. So again, your mileage may vary. You do need to play around with these settings. And then that then moves on to the bit more sort of relaxed things that you may need to play around with. So save to Google Drive. If you're using your Google account, it will save the data, the structures, and also all of the images to your Google Drive. And also how high resolution do you want to export the images. Once that is done, this block will take quite a while. And then run prediction. So then that will start the prediction process. And once that is complete, you can then display the 3D structure if you wish to see so, see the structure in the workspace, but they will all be deposited into this little folder on the side here. And also, if you've checked the Google Drive setting here, it will be deposited into your Google Drive as well. Okay, yeah, and then at the end, you press package and download results. That will download a zip file into your local computer. And that is pretty much it for AlphaFold. It's a pretty straightforward process. It is quite newbie friendly if you just would like to dip your toes in AlphaFold, and you're not quite ready committed to dedicate half your lab to working on this. So that's pretty much a rundown of AlphaFold. Is there any questions in chat? Like I said, feel free to play around with this in your own time, or later on, if you completely get lost in the Nathaniel's walkthrough, you're always welcome to come back to this and play around with this. Yep.
I have a question, Jake.
Yep.
So yeah, you mentioned if you get bad results, like how do you know something's a bad result or a good result?
Okay. Yep. So that comes to the next part of my talk, which comes to so there are a few quality metrics in AlphaFold. So there are the AlphaFold provided metrics, but there is also metrics that you provide that you can look at yourself. So I'll go through the AlphaFold metrics first, and then some of the methods that you can also use to look to see whether your structures are good or bad. So let me just screen share the next, which I'll have to pull up. Give me a second. Okay. Actually, give me a second.
There was one question in the chat about the size limit.
Size limit for Colab. So Colab, for the free service, I think you can go up to, I think, 500 to 600. You will have to test it yourself because it's sort of like a black box. Some residues take up more memory. It's all memory bound, so memory on the graphics card. So the VRAM of the graphics card. So for the free service, it's quite limiting. Whereas for the paid service, it's a lot better. So you can get up to, I think, a P100, which allows you to predict, I think, just over 1,000 or so, up to 1,500. depending on your luck. That's sort of where it maxes out. So if your protein is bigger than that, you may wish to use one of the other services, such as running it on Artemis or running it on, yeah. So I tried to run it on Colab, we've got a module not found error, no module named bio. Okay, so is that during the install dependencies part, Ghana?
Yeah, it sounds like you may not have run the install step, maybe.
I think, yeah. So I think you'll need to go through everything in order. So the inputs protein sequence, you need to run that and then install dependencies.
I thought it might be worth noting as well on Google Colab, you can you randomly get assigned a different GPU. So yes, sometimes it'll work, and then another time you might find yourself on an older and different version GPU, so your sequence won't work anymore. So yeah, just keep that in mind. The resources are a bit unpredictable with Google Colab.
Yeah, so that's why if you like something that's a bit more reproducible, it may be better to, yeah. And I think Lagana, because I think no module named bio, I think that might be bio Python. So I think you might have got to install the dependencies, or the installation of the dependencies not finished yet. Yeah. Okay, so I'll just share with you some things from my thesis, which might be of interest. Okay, so this is what's called a PAE plot. So it looks like, like a tartan, sort of one of those Scottish tartan things. And so what this shows is on the x-axis is the scored residues. So that is the predictions that AlphaFold has put in. And then the aligned residues is the real protein structure that it is comparing against. So in this case, it's comparing against one of the experimental structures, and the scored residue is the structure that is generated by itself. So it goes from dark blue to very, very good alignment to yellow for very poor alignment. So this is useful to make sure that if your protein has certain domains, so in my protein, it has, transmembrane domain and also intracellular domain. So in the cases of these, different domains should line up to each other. So you can see that this domain here and this domain here, if I got a little pointer, so this domain here, oh, that's a terrible color, red. So this domain here and this domain here. So these are two separate proteins. This is a multimeric prediction. So even though they are the same, they are the same domain. However, even though they are not the same protein, they still align very well with each other. And so, yeah, so that's a good measure that your protein prediction's done pretty well. If you're expecting to see a domain level similarity, but it is not present, that probably means that your protein has not, been folded correctly. Okay. And the next metric, which is also very useful to use, is the PLDDT plot. So PLDDT is a value which basically shows how well that area of the protein lines up with the comparison structure. So you can see here that the very, so the domain, so the extracellular domain and then also the intracellular domain here. So most regions of the protein have very high similarity. There are large dips here. So one dip in particular, in my case here, so this very large dip here, so that one corresponds to a very large intracellular loop, which has previously not been characterized before experimentally, simply because of how flexible it is. And so that is a very useful metric of telling, so if you're expecting a lot of flexibility in that region and AlphaFold's showing you that flexibility, then that's a very good sign. But otherwise, you can see that the score hovers around 90% roughly for most of the protein, and 90% or 85% or above is considered very good prediction. So I would be concerned if your output is showing us very poor for most of the protein. Okay, and then I will show you an example of what a protein would look like. So here is a PyMol entry. So here is a predictor protein which I have generated for my work. And so you can see here that the protein, so it looks, this is the final protein which we've generated. So we've generated 20 structures in total, and we rank them and we perform energy calculations on them to determine like molecular mechanics calculations on them to determine which one is the best. And this is our top model. And the good thing about AlphaFold is that the PLDDT values, which you saw previously, it is actually written as a B factor inside the PDB file. So what this lets you do is that when you go to the color, you can actually color by the B factor, and it will show you which regions of the protein is flexible and which is not. So you can see here that most of the protein, which is stable, so the transmembrane domain, and also most of the extracellular portions of the protein, you can see that it is very stable. And so those parts are the high confidence regions. However, you can see up here, these protein loops up here, these are known to be flexible in the literature because they correspond to drug binding sites, well, allosteric binding sites. And so we can see that in this one here, this is a very flexible region. Moving down, we can now see the very, very flexible regions previously discussed. So yeah, the color scheme, so red is very good, and then goes down to blue for very, very poor confidence. And so confidence is a metric of the flexibility, essentially. So you can see that these intracellular loops here, they are very, very flexible. And so these have not been resolved previously using experimental methods, simply because they would just flop around and they're just impossible to characterize. But we can visualize their lowest energy state using AlphaFold. And so, yeah, so that's just the visualization of the parts of the protein. And so, yeah, that's pretty much it. Is there anything that I missed, Nathaniel?
I think that's pretty good. Did anyone's collab complete to succession?
Yeah, mine did.
Good, it's good.
Sounds good.
Mine as well. Cool.
Yep. Yeah, but yeah, so that's it basically for AlphaFold itself. And now I sort of go off the track a bit to talk about like what else you can do with it. Oh yeah, so the metrics that I was talking about earlier, how do you know it's good or not? So another thing, a very basic one you can do is that if you're predicting something that is already have a previously determined experimental structure, you can simply run a superposition and see whether your protein and that protein is, comparable. And if it's completely different, then something's gone wrong, and you may need to tweak some settings. If you're working with a peptide, then that may be a little bit more difficult. But yeah, and another method is that AlphaFold is not it's not a end all and be all. So it's used to supplement other methods as well. So don't think of AlphaFold as a method which basically, okay, well, I'm going to run AlphaFold and that's my research done. So it is a method used to feed into other methods. So I've currently used it for free energy perturbation, so calculation of drug binding affinities, using the AlphaFold models to dock drugs into, and then compare against currently available data, and then running FEP or other alchemical methods to determine drug binding affinity. And then also some other methods such as molecular dynamics as well you may wish to consider running. So so I've ran a lot of MD to validate some of our data and compare them to experimental data when running under the same MD parameters, and see whether the proteins behave similarly in regions which are similar, and how differently they perform in regions which are different. So there's a lot of methods, and also you can use it in your cryo-EM studies as well. So fitting into those cryo-EM maps, if that's what you're interested in, and also a whole bunch of other studies. So it's a step which enables you to perform other research. It's not so much a method which you can, just predict 10 protein structures and then publish that. Because there was a phase last year where there was a lot of that, and then people sort of realized, oh, that's not it's not as simple as simply just dumping things onto into a paper, and then yeah. So I think there's a few questions. So the protein that you showed seems bigger than 500 to 600. Yeah, so that one was actually predicted on Artemis and also on Gaudi as well. So that one required the use of two V100 GPUs. So yeah, so that one required two V100, and some later models on Gaudi use the newer A100 GPUs because they have a larger VRAM capacity. So yeah, so these are not done on Colab. So Kiva had a question regarding the energy of molecules and molecular mechanics, if you have specific software or code. Yeah, so there's a few that I've used. So Schrodinger suite, yeah, but I'm happy to drop my email. Feel free to email me after, and yeah, I'm happy to have a chat about that if you're like more guidance on that. With a version of the PDB database that Colab is using. So the collab should have, yes.
It's
So
on the first line of the documentation. That
yep,
what version it's using.
that's right. Yeah. So PDB, so AlphaFold 2, the training database that has been updated to a newer version as of December last year. So I've had a chat with the actual developers at DeepMind, and they said that it wasn't really necessary because they didn't get any performance improvements. But academics, being academics, they were complaining too much about the database being old. And so they did it just to keep people happy, basically, but they don't see any performance improvements. So, yeah. How does AlphaFold work with proteins that don't have known reference sequences? Does it struggle at the MSA step as the sequence is very divergent? Yeah, so it actually performs very well. So this is not first-hand experience, but when I was in America at the conference, so I've seen groups where they use essentially proteins, essentially sequences with less than 5% consensus to anything that we've currently really got available. So it's from a weird and wacky bacteria, which was isolated from Antarctica, I think, which was evolutionary divergent from the rest of the world for, I don't know, I think it was like a long time. Yep. And then so it had less than 5% consensus with anything that we've currently, know of that's in the database. And they used AlphaFold to predict a certain protein in it, like a metalloprotease, something like that. And they used that to predict it in AlphaFold, and then they obtained cryo-EM structures. And the results were surprisingly close. I think it was more than 99% similarity. Given that it's essentially got no consensus with any other living thing in the world, you know, because it's been so evolutionary divergent. And so, yeah, but obviously they played around with the settings a lot, and they actually worked directly with DeepMind on this project. So there's a lot of back-end modifications. So like I mentioned earlier, it's not a plug-and-play solution. You do need to play around with the settings, which is where the time, which is where a lot of your time would be spent. So yeah, getting the model itself is the easy part. It's actually tweaking those settings and optimizing the parameters. Okay, I think. All right, Chandrika has a question. In your experience with the P2XR family, do you see that predictions are better when these are experimentally determined structures? In your example, there is a rat cryo-EM structure published. Better when experimentally determined structures. So, like I said, it's not. It doesn't need to have. So obviously, if there is experimental structures available, it does improve the scores a lot because it does help with it a lot. But for our use case, we were actually looking at novel variants which have previously not been characterized and which were impossible to characterize using homology modeling. And so, yeah, so some sequences of the P2XR variant we looked at has no correspondence with any of the currently available P2X sequences. And so, yeah, so essentially, yeah, so that's the main difference. And like I said, the example I gave, you don't really need experimental structures determined in order to improve these scores. It does really well by itself, but you do need to spend the time to optimize these settings. Yeah.
That's great to know. Thank you.
No worries. I'll just drop my email in the chat. And I think Nathaniel's got it as well somewhere. Yep. So yeah, so if you'd like to, send me an email afterwards, with any questions or whatnot, yeah, I'm happy to answer those. And yeah, and then I think that's it. Is there anything else I didn't cover, Nathaniel?
I think that sounds pretty good. Yeah, but maybe we'll take a 10-minute break and then we'll come back and connect to Gardi. I mean,
Yep.
sorry, Artemis. So and we'll go from there.
Sounds good. Yeah, and then since I am finishing my PhD, if any of you is looking for a postdoc to do this stuff for you, I am available. So if you want to hire a postdoc to do all of this for you instead of yourself, yeah, let me know. Cool. All
Good
right.
stuff.
Thank you.
All right, we'll see you at 11:08, everyone. All righty, everyone. Let's maybe wake back up, and we'll get stuck into perhaps a more technical part of, or a different kind of technical part of the workshop today. For this, we're going to connect to Artemis and see if we can run a job on that, and hopefully we'll see kind of some of the difference between AlphaFold, which Jake demonstrated, and the native AlphaFold, and then also you might be able to explore how we can kind of automate things. So Jake was talking a lot about parameter tuning and doing a lot of trial and error experimentation. So this is where sort of the command line can really come into power, because you can just queue up 1,000 different scenarios and click run, and then come back in a week or so and find the results, instead of sitting at your AlphaFold and clicking next. So yeah, we'll see a few different strategies and a few different tools to use. But let's get going. Alrighty, I will share my screen again, and I might just let's do this. Yeah, okay, so I'll share this portion of my screen. So a lot of what Jake spoke about, I think, is in the some of the notes here. We'll also get him to add the presentation slides to this site, and the links off to the various collabs and a couple of other collabs. Slight different variations are in the notes as well. But we're going to pick up. Actually, we'll pick up at the setup pages first. So that'll be the first one that I link. So I'll put this in the chat, and I'll just kind of talk through some of these setup instructions that hopefully people have done. We have spare Artemis accounts. So if anyone doesn't have an Artemis account, we can provide one for you for today. But it probably would be best if you could work in your local account. But please maybe just post in the chat if you need an Artemis account. If you don't have one, you can get one, not immediately. It takes like a day or so, but at Dasher. So that's the Sydney Research Dashboard. So I've just clicked off that link. Hopefully it's loading. Thinking about it. Try go there directly. There we go. Maybe that link was not working. But anyway, hopefully everyone is familiar with the Research Dashboard. If you're not, especially maybe if you're a student or something and you haven't seen it before, I'm just doing two-factor authentication on my phone here. Okay, I'm in. All right, so this is the Research Dashboard, and this lists all the various projects that you're part of. You might just be part of one. I'm part of many. And so in here, you can click on any particular project. It's a reasonable one. So here we go. I can click on part of the SIH data science team, and I can scroll down here. So this is all the people on my in this project. But down below all that, maybe no, I missed it. Sorry, up. They're all the changes that have been made over time. So down below the list of researchers, this thing called Research Services, and you can see that this group is part of Artemis. So that means we have an Artemis account associated with this group. So everyone's UniKey who is part of this project will have an account on Artemis, and they will have access to this particular project. So I'll talk through what that means. Okay, and if you don't have that there, you can either modify the project or you can just create a new one as well. All right, so feel free to ask any questions about that. So that is the Research Dashboard. I can put that in the chat. So let me go. Awesome. Okay, so that's the first thing. If you haven't done that, yep, I recommend it. But if not, you can get a training account. But please just indicate in the chat if you need a training account, and we can do that. All right, so I speak about something called singularity. I'll talk about that later on. You don't have to do that now, especially if you don't know what that is. The next thing we need to do is get a terminal client. So I'm on a Mac at the moment, and so this has a built-in terminal client. If I just search for it, terminal, it will open up. Oh, there's one I prepared earlier. Let's minimize that one. And so this is the Mac terminal. I'll just zoom in so we can see. And basically, once you have an Artemis account, you can connect to Artemis. So with using something called SSH. So on this terminal, I can navigate around my Mac computer. I'll demonstrate Windows in a second. But on Mac, I can type in SSH, which is a command. I can give it this option called -X, which I'll explain later. And then I put in my unikey and hpc.sydney.edu.au. So that's it. So I put in my unikey and also the address of Artemis, which is hpc.sydney.edu.au. Keeping in mind, I'm currently connected to the VPN. So I'm not on campus, but I am connected to the VPN. So that's required for Artemis. And then I put in my my unikey password, and this should connect me to Artemis. Hopefully. Thinking about it. There we go. So there's always like a little note on about what's happening on the on Artemis, but I'm now on a remote computer. This is based out in Erskine Park, Western Sydney. And yeah, this is the university supercomputer. So I can navigate around this computer just like I would my local machine. But we'll get to that in a second. Now, if you're on Windows, there's no great built-in tool. Actually, I don't think there's any tool that uses what's called the SSH, secure shell. It's called the protocol, the secure shell protocol. And so instead, the easiest thing to do is download this tool called Putty. And so Putty is just a nice little graphical user interface. So I just clicked on that Putty link. I can post this one to take you straight there if you need it. I will talk about WSL in a second. So you can download an installer, or you can just download the alternative binary file for probably most people would be on x86 64-bit. So if you download that, it's just an executable. And so in theory, you can just run that. You can run that on any machine, even if you don't have permission to run on it, like a lockdown hospital computer or a university machine, like a lecture theater or something. So it's like you can run it off a USB stick, so it's standalone. So that's quite handy. Although I'm on a Mac, so I can't run it. However, I have got a Putty demo, and so once you download that, I will then. Where is it? Okay. There's my here we go. So I have a version of Putty. It should look like this on Windows. So basically, you launch the application, and then Putty looks something like this. And in the host name, I can put in hpc.sydney.edu.au. There's a whole bunch of configuration options that you might require at some point in the future. But by default, that should be enough. You put that in and you click open. And then it will open up a new terminal. And it says login as. And so I can put in my unikey. And then it will say, and your password. And you'll notice that when I type, it looks like nothing's happening, but that's a secure feature, not a bug. So I press enter, and then I'll have the same experience that I have on a Mac. Yeah, so that's the Windows version. Let me bring back some. That was me installing Putty on Windows. This one. There we go. Okay. So yeah, there are that is one option for an SSH client on Windows. There's a bunch of different other ones. One that I highly recommend, and Lagana has recommended in the chat, is WSL, and you can get like a full Ubuntu installation, so a full Linux installation on Windows computers now, and it's pretty easy, but it sometimes has issues setting up. So that's why I kind of don't go through it, but I'd highly recommend that, especially if you're doing more Unix type stuff as you automate things. It can be really, really helpful. All right, so connect to that, and then and that's it. So that's basically our setup instructions. We'll download some data shortly, and connecting to Zoom, hopefully. Connecting to the VPN. Hopefully everyone knows how to use that. Okay, so I'll go to sessions and talk about Artemis a bit. I'll just talk through this. Are there any questions? And has everyone connected to Artemis, or has anyone not connected to Artemis? Cool. Okay, that's fantastic. Okay, so yeah, Artemis is we've got about 7,000 CPUs, 45 terabytes of RAM that you can use a large chunk of. Lots of, or not lots, but we have quite a few GPUs as well, and we got them, you know, when they were brand new. So these things are kind of starting to age. I think we've got more storage than that. We've got a petabyte of storage now. And yeah, it's basically what's called a cluster computer or a supercomputer, and it's really great for long running jobs. As we've seen with Colab, Google Colab, you know, you have to click buttons. You can't really do that on Artemis, or not. It's not designed for that. It's kind of, you click run and then you let things run for a while. You can, but the beauty is that your internet connection can go down. You can turn your computer off, and it'll all run there. And you can use huge amounts of RAM and use lots of data input and output as well. So if you need lots of data, so I'd imagine that you'd use AlphaFold in sort of a larger workflow where you'd maybe make predictions and then use those output PDB files into something else. Okay, and yeah, please acknowledge us if you do use Artemis. All right, so I believe everyone connected. So let's jump straight into this one, and as I talk through this, I'll explain some of the ins and outs of Artemis as well. All right. So I am currently connected to Artemis, and if I type in the words PWD, or the letters PWD, it says I'm at /home/nbut3013. So this is my local folder. This is kind of like your home folder on Windows or Mac, where you'd have like your documents, your downloads, those kinds of files. And on a Unix environment, the slash, I guess, is equivalent to maybe like the C drive on Windows. So anything starting from there is like the root, what's known as the root of the computer. I can issue the command ls, and this lists out all the items in there. So I have a lot of things in my home directory, and I'll talk about some other commands as well. And in fact, I'll do that right now. So we're in this directory, home/nbut3013. In your home folder, you only have 10 gigabytes of storage, and so that can really fill up pretty quickly just with configuration files. If I do ls minus la, ls minus a, there's all these hidden files that have a dot in front of them, and so things like Wolfram and like .ssh and .singularity, all these things, they generate default configuration files, and often they blow up in size. So you can fill up your home directory very, very quickly. So I recommend not generally working in there. Instead, we're going to work in another folder called project. And so this is storage which is shared between everyone in your project. So I'm going to navigate to cd /project slash training. That's where I'll work today. This is a project that I'm part of. And if anyone needs to use training accounts today, they can also use that same folder. And so what I just did, I typed in CD, which stands for change directory. And then I put in the name of the project and then the name of my project. Now, if you don't know the name of your project, you can type in, what's the best way? There's quite a few ways to do this. One of them is called job stat. And this will list out, it tells me all my jobs running, which I don't have currently any running. It lists out my home folder and says how much storage I'm using in there. So I'm using 7 out of 10 gigabytes. And then it lists all my projects and the storage that I'm using for each of those projects as well. And the way these are kind of broken down is it starts with kind of like your faculty identifiers and it ends with this RW. But the only part that you actually need is the middle section, and that's called your project short name. So the one that I've just connected to is this training one. So that way you should be able to see which projects you're a part of, and then you navigate to it with CD/project / and then the name of your project. Okay, is that clear for everyone? And that command job stat is it's an Artemis specific command. There's a few other commands as well. You can type in groups, but it's just not as pretty. You get the list of groups that you're a part of. That one's a general Unix command. And I think there's a few other ways to tell which groups you're part of and how much storage you're using and that kind of thing. But for now, yeah, job stat is a handy little tool that Artemis has. Okay, so where am I? I'm PWD, I'm in /project/training. If I do ls here, there's probably a bunch of directories created by people who have used the training project in the past. And for today, I can make a new directory. So I might just go make dir, and I'll just call it AF demo. So AlphaFold demo. So, and then I'll go CD AF demo. Okay, so that's, you do not have permission to be in the training account. If you don't have your own UniKey account, if you don't have your own UniKey account, we can give you an account. Otherwise, you can do this in your own project folder. Yeah, so check the name of your job, your project with the job stat command to see what project name is, and then do your equivalent /project/ for me, I'm going training. And then I just made that folder called or you can do that CD/project/ whatever yours is, and then make the AF demo. Mine will say that already exists because I already created that one. So then you can change directories into AF demo. Okay. So the next step, so if I type PWD, it says where I am. I keep repeating myself. That's intentional. So if I type PWD, it says where I am. If I type ls to list out everything in there, there's nothing in that folder. We haven't created anything, but we're going to fill this with some data. So on the training documentation here, there's this command that says how to download the data. So you can execute this command. I will post that in the chat as well. And actually, if you're on Party, how do you? Hey, Mac is pretty easy to copy and paste in the command line, but on Party, I always forget how to do it. I think you can copy as normal, and then if you right click, it should paste the command into the command line. But basically that should download a zip file. You can also just download it locally as well, and have a look at it. And I'm just going to unzip it as well. So I do unzip af demo.zip. And if I type ls, I can see it's created this new folder called af demo. All those files that came on screen, they are all the files that are in that zip file. And if I go cd af demo, I can see I'm in a folder now called /project/training/capital AF demo, and then /AF_demo. Okay. So yeah, you can copy from the notes there. It's basically wget, unzip, and change directories. So that's what we have to do. And then in here are a handful of different files, which I'll talk to in a second. Does anyone need help with any of those steps?
Yeah, hi Nathaniel. I was just wondering, could I get access to one of those training accounts? Because my login got set up very recently, and it seems like I don't have access to set up new directories and download the GitHub repository.
Okay, you can be. This is very generic. And actually, I can share this with everyone because we update these frequently. So the username is ICT_HPC train, and the password, let me get this right. Okay, so yeah, the username is, you can use ICT_HPC train 2, but yeah, it goes 3, 4, 5, 6, etc. So if anyone else needs one, use a different username. 4, 5. And they have the same password. Sorry, Lei, what do you mean? Can I type where? Oh, I've posted it just above your message. So the username is ICT_HPC train, and then a number. And yeah, true. It actually won't matter too much if people use the same account. But yeah, maybe pick a random number between 1 and 40. Okay, good stuff. And yeah, once you log in, so I'll just redo that. I'll actually do it in a training account. So I'm just going to exit. Okay, I'm going to close down my terminal. Close that one down. Sure. I'll start a new terminal. Oops, didn't start a new one. Yeah, start a new terminal, and I will just run through that one more time. So I'm going to SSH -x ICT_HPC train. I'll do number 1 at hpc.sydney.edu.au. I connect with the password, in this case, sad green caps 678 exclamation mark question mark. Okay, and then so I'm now logged in. If I do ls, I can see this account has a bunch of different items in there. If I do pwd, I'm in /home/ICT_HPC train 1. And then finally, if I go /project/training, and I can make a new directory, make the maybe call it let's call it AFNat. So I know that it's my one because we'll probably have a few AF demos there. And I go cd AFNat, and I then can say pwd, where am I? I'm there. All right, and then I'll download that file using wget. Okay, that's listed there. I will unzip it and I will change directories into it. So I am now in /project/training/afnat/af and in there are 5 different files. So they should, I'll just kind of post, once you connect, post each of those commands in the chat. I don't have time. Make sure you call it something else, especially if you're in the training project, because you will get an error saying that folder already exists because it belongs to me. You get unzip. I'm just putting these in the chat one by one. So I think if you log in and actually just run them all, it should work. And then we're all up to the same spot. Okay. All right, good stuff. So yeah, if you're still having troubles, please post in the chat, and I think Chris or Nandan can help you out. Now, what we have here in this directory, firstly, there's a fasta file. I can do something like more input fasta. It's just a text file, so that just prints that to the screen that I can see. I can actually even create one because it is just a text file. I can, just make a dummy one. So dummy to demo.fasta, and then I can echo the sequence. With 2 colons, with 2 chevrons. And so, yeah, it's like I said, it's just a text file. You can get that onto Artemis however you like, either generate it straight on Artemis, copy and paste it into a Word document. You can use something called Nano, which is a text editor. Hello, here is my fast day. And yeah, you can put in the sequence there. I won't do that now though. So there's lots of different options, but basically that puts it there. And then once you have your FASTA file there, what we're going to do is submit what's called a job to Artemis. Now, because Artemis is a big shared system with, we have about like hundreds, maybe 200 unique users each month, and maybe 300. And all those people are sharing those, even though we have 7,000 resources. Yeah, even though we have those resources, you still have to, it's still a shared resource. So that means that we have to use something called a job scheduler. And basically we say, hey, Artemis, we would like 10 CPUs, 100 gigabytes of RAM, and we want it to run for 2 days. And it says, no problem, there's a space opening up over here in 3 hours time. I'll put you there. And meanwhile, someone else is like, I need 1 CPU for 1 minute. And it's like, no problem, your job can start immediately. And so it balances out the supply and demand, basically, of what's available, what's not. Leigh, I just got a message from you. So maybe Nandan, are you there?
Oh, yes, I'm here.
Can you maybe I'll just make a breakout room. How do you get out of Nano? Sorry, I tried to escape out of Nano quickly because I realized that might be a problem. I press Control X. Okay, wait, I'll just catch up with these questions. So the first one from Leigh is that you can't log in. The PuTTY wouldn't allow you to type it in. So it might be actually working and it just doesn't show up on your screen. Like it looks like nothing's going in. So, but let us know if that's not the case. Now, how do you get out of Nano? Did you get out of Nano? Okay, good stuff. Which project to use within your own project? Yeah, that's a good question. So, I mean, you can use it. You can use any project. You might have particular tasks. I'm part of like 15 projects, and I have one or two that I use mostly for specific things. But if you're doing work with certain collaborators, then they may want to be able to work with you on that data set. So it makes sense to work in that project. It doesn't really matter, like from a technical point of view. But yeah, from a research integrity point of view, then you may want to work in the same project. Even if you're part of multiple projects, you can kind of mix and match from a technical point of view. So keep that in mind. And yeah, keep in mind everyone in the training project has read access to all your data in the or somewhat some amount of read access to all the data in this project. So and yeah, I haven't spoken about Scratch yet, but Scratch is another place. So project has 1 terabyte of storage. Scratch has essentially unlimited storage, although in practice it's more like multiple terabytes for any particular point in time, up to anything up to about 10, and you don't have to worry about it. More than that, you might consider doing that. Access denied. Sorry, I'm just looking at this image here. Oh yeah, you guys can see this. Are you sure you're putting in the right password? I guess that's that would be my questioning for that one. So did I make a typo? I don't think so. I think I used the same. Does anyone else have success with the PuTTY password? I'll just okay, that's good to know. Thank you. All right. Sorry, any more questions? Okay, the password was posted up above. Thanks, Nandan. Good stuff. So yeah, just post generally in the chat, and I'll keep pushing ahead for most people that have access. It shouldn't matter if multiple people are using the same account, but yeah, try using a different ICT HPC train account 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, and you do need.
So Nate, is it only up to 20 or something? I just gave someone
Up to
20. 40, right? Yeah. Okay.
1 to 40 inclusive.
Okay.
Yeah. Okay, and we'll have a break shortly, so I can talk through anyone that's still having issues. But I'll just persevere for now anyway, and you can just follow along. So what we're going to do is make what's called a job script. I mentioned that the Artemis is built on it's a shared resource, and so this script here, it's basically what's known as a bash script, which is like a recipe of instructions to send to the computer, and that's how it works on Linux or Unix. You can do it on Mac as well. Windows, again, a bit different. You can write scripts in Windows as well. But yeah, it goes from top to bottom, and it tells the computer what to do. Now, this one's a bit of a special one because it has these what are called PBS directives, and they're specifically specific information for Artemis or for the job scheduling software we use called PBS. And so what that means is that this is where we say which project we want to run it under. We say how many nodes, how many CPUs, and how much memory we want to use, and how many GPUs. That's an important line. And then how long we expect it to take as well. So we set the wall time, and then you can give it a name. There's quite a few more options. I won't talk about those, but yeah, there's a whole bunch of other options that you can include that let you kind of control your job in more advanced and amazing ways. But we have trainings on those as well. Okay, then the next thing we do is we load in all the software that we're going to need. So Artemis has sort of thousands of pieces of software installed, And if they were all there just straight away, they'd all conflict with each other. We have multiple versions of the same software. And so what we do, we use this tool called Module to load in just the software that we need at the time. And we can see that AlphaFold is one of those bits of software. And you might be familiar with these other ones, HMMER, HH Suite, K-Line. So AlphaFold actually uses these under the hood to do a lot of the work that it does before it actually gets to the sort of the new part, the new AlphaFold predictive stuff. Okay, and for the full list of those, you can go to here. Actually, I say the full list. It's probably, it gets outdated pretty quickly as new software is installed. Software list. So I'll just post that in the chat. And so you can see all this different software that's listed. Actually, I wonder if AlphaFold is, oops, AlphaFold. Oh yeah, AlphaFold's not even listed there. So it's somewhat, yeah, it's outdated, this list, but it gives you an idea of what's available. Okay, then the next thing we do is we have to change directories. So when you normally log into Artemis, you land in your home folder. And so you generally don't want to work in your home folder. Like I said, you want to work in your project folder or on Scratch. And so this little command says, change me to the directory that I submitted the job in. So this PBS_O_workdir is like a shortcut to say, change me to whatever folder I was currently in, which for me will be project training AFNAT, AFDemo. Okay. And the next thing is we can set some little, we can just set like variable names. So here I set a little out directory, so somewhere that AlphaFold is going to save our data to. So this can be anything. It can just be the name of a normal folder, but I use a little bit of scripting magic, so we get some unique folder each time, and then it creates that output directory as well. All right, then importantly, now AlphaFold uses a lot of databases under the hood, and they are located on Artemis because they're fairly large, sort of terabytes in size. There's this shared data directory called project/data, and in there, I think there's quite a few sort of different databases, but there's one specifically for AlphaFold, AlphaFold 2. And it has a few snapshots of the databases. So it's got a 2022 snapshot and a 2023 snapshot as well. And so we just have to point to that directory. So AlphaFold knows where to find the various databases. You can install your own databases on Scratch, but it would probably be really inefficient. A better way, if you do actually you need specific databases, is to log a ticket. And I think that's in the documentation here. Log a ticket and say, hey, I really need this support, this database. And then they'll say, oh yeah, maybe, or we're out of space or something like that. Okay. All right, but yeah, then this step, so after we say where the database is, this is how we run it. So this is how we run AlphaFold. We say run alphafold.py. So when you type in module load AlphaFold, you'll get access to this run alphafold.py command. And then we give it a list of different variables. So most of those kind of fixed because it's just all the link lists, the locations of the databases. The key one is your input faster. So you can specify that. And then you set your output directory. Then yeah, database. And then there's a few options here that you can now change. Now all these, the full list of options, AlphaFold, is in the documentation of AlphaFold itself. And that's really the only way to know what's exactly available. Yeah, they have basically in here are all the different explains what options there are, how to use them, and how to execute them. So I can post that in the chat if anyone's.
Nate.
Yep.
Can I interrupt you for a minute? I think a couple of people are having some issues with the logins.
Okay.
And they said they are on campus and still the password is not working for the
Password is not working. Okay. Let me just try again. So
I'll try one from my side.
So I'll do it via Putty.
Yeah, they are Windows people.
Okay, so I type in hpc.sydney.edu.au and click open. I log in as ICT_HPC train. I'll go 11. And then the password is SAD green caps 678 ! Question mark. I don't know. Beyond, typing it in properly. How about, we'll like I said, we'll have a break in 5 minutes, and we can get everyone sorted then. Because once you're logged in, it should just be a couple of lines to get back up. So yeah, I'm not quite sure. There's often a few things that can be going wrong.
I think the password that's been typed in is actually different to what you just typed in. The exclamation and the question mark, if that's like, if you typed that, then that wasn't in the chat.
Oh, really?
Yeah. I think that's why people might be having problems. There might be a difference between the password that's been typed out.
Okay. Should be this one.
Yeah, it's different to what's been typed out previously. I think it was just missing the exclamation mark and the question mark, so that should work now.
All right. Okay. Very good. Good stuff. Thanks for picking that up. So hopefully that should be okay. All right. Okay. So if we, okay, I can jump back in then if people can connect with the updated password. Good stuff. Where is my AlphaFold? Oops. Okay, yeah, so you have all those different configuration options, and you can do things like, this is one script, but you could submit 1,000 of these very sort of simply. We won't go through kind of how to do that, but you can. So that's sort of where the power of this will come into things. And so this script is already prepared for you in I think it's called the alphajob.pbs. So you can just dump that to the screen by typing more alphajob.pbs. You can see everything that's in there. So it should look pretty similar to what's available. Actually, it does it a little bit more fancy, so you don't even have to link to any particular folders or files. I think I just generate the input FASTA immediately on the path like that. Okay, and so if you just run this, it will take about 40 hours to complete. So I'll explain, why that's the case, I guess, and things that we can do to speed it up. So, but if you just run it using these default configuration options, which doesn't use very much RAM, I think that's the main thing for this particular sequence and these set of options. Then it will take you around 40 hours or so to complete. But we can do that. We can submit a job to the queue. So if I just type in q sub alpha job.pbs. Oh, it got rejected. I know why. Okay, so we're gonna have to modify this. So this is because, oh, sorry, was there a question? Okay. Good stuff. Yours in the queue. Fantastic. So if you're on a training account, it probably will be rejected by all possible destinations, but that's okay. So I will talk about Nano now. So if I type Nano alpha job, alpha_job.pbs. So in here, you can navigate around using your keyboard. I'm just using my keys to navigate around. You can specify the project. So I'll keep it as training because I'm in the training account. So that's the only one that I'm part of. Now here I set the CPUs and the RAM and the GPUs. So it's currently set to 0. And the wall time, now the wall time on the training accounts is actually only 24 hours. So it's restricted. The main queue is much longer, many weeks, I think is the longest you can run jobs for. So if I just set this to, let's just set it for 10 minutes for now. And that's it. So you can make any other changes that you'd like to change. And then I'm going to press Control X as indicated down here. It says press Control X to exit. That's Control X, not Command X if you're on Mac. And it says saving the modified buffer will destroy changes. And I say, yes, please save. And the file name to write to is alpha_job.pbs. And I press enter. And I now have an updated alpha job. And so I can type q sub alpha_job.pbs. No such file or directory. Okay, now I've made a new mistake. What did I do this time? Script file, no such file or directory.
Or the file name.
Oh, I typed q sub alpha_job. Alpha job. I did it again. Job.pbs. When you get an error on Artemis, I think 90% of the time it's probably a typo. So keep that in mind. All right, and now it's submit to the queue. And we get a little queue job number ID, a job ID there. I can type qstat, and if I type qstat, it will list all the jobs currently running on Artemis, or in this case, it will run all the jobs currently running in the training node. But if I do qstat -u and then my uni key, or in this case, my HPC train, my train 1, it will just list my jobs. And I can see, yeah, we've got this job. It's in the training queue. So yours might be in the normal or the short or something else. It's got the name of my job, how many nodes and CPUs and RAM I asked for. And it says the status here is running. So the status might be queued or held or something like that, and how long it's been running for. Okay. All right. Yeah, if you do need to modify the script, so using Nano, you can go in and change this first line. So this is that first line is actually the only required one when submitting a job. So you have to set this to whatever the name of your project is. So I have a project called SIH Next Gen. Yours might be something else. So, and if you don't specify a job, Artemis will default to what's called the default queue. And that's basically your only option anyway. So unless you're doing something fancy like an interactive job or data transfer, I think they're your only options: data transfer, interactive, and the default queue. And the default queue has all these sub queues: the small, normal, large, GPU, high memory, but you can't submit that to them directly. So yeah. Okay, so once again, yeah, you can change this -p option, and I press Control X, and will I save it? Yes. And the file name, alpha job, and submit it. Okay, I'll put another 10-minute break on, but feel free. So we won't do anything new for 10 minutes, but if you're having any issues, I'm happy to stick around and help people. I'll even pause the recording. So I'll see everyone else at 12:07 for resume sharing, and we'll get stuck back into it. So yeah, we'll keep helping out people as they need it. Well done, everyone. You know, this is I know there's a bit of a mixed crowd of people here. Some with more advanced HPC skills than others. Keep in mind, once you can connect to Artemis on this remote computer, you can basically use any Linux system. You can connect to any remote computer. So yeah, knowing how to do this is really handy, and not just for AlphaFold, but for any tool that you might need on a system like this. Okay, so what we've done is basically log in and download our data and set ourselves up. So I have done that here. So I'm currently in my my project training AFNat AF demo directory. I'm using a training username unikey, which is part of this thing called training, this project. If I type job stat, I can see the list of projects that I'm a part of. So this particular unikey is part of a handful of projects. So I can use this short code. You probably know the short code of most of your projects if you're familiar with -R and the RDMP process. All right, and I can see how much storage I have available. So this project has 264 gigabytes out of 1 terabyte used in that folder. All right, so now what we have in this directory, I've got a few new files because of that job I submitted. So I'm going to look at just running that again. So I'm just going to type nano and then this script name, which is alphajob.pbs. So again, this is just a text file. You can write this completely from scratch. You can write it offline and upload it. There's a few different options, but basically, once here, the only really required point is specifying the project you want to run it under. So you could, for me, I'm running it under training, but you should replace that value with whatever the name of your project is associated with your unikey. Okay, then everything else. Also, if you're on the training accounts, you actually had to make the wall time lower because 96 hours is too long for the training nodes. It's like a special secret queue. So I just put it for 10 minutes. So my job would run for 10 minutes. And then basically after 10 minutes, it's going to break. It'll just it's like pulling the plug and turning the computer off. So keep that in mind. Now, so after we set our PBS directives up there, we load in the software that we need. So in this script, I've loaded in something called CUDA, which is used for GPU stuff. So it's a little bit different to the notes. And the other thing is this, the AlphaFold version. So we only have AlphaFold 2.2.2 on Artemis. I think newer versions probably won't work because Artemis is pretty old. So its underlying operating system can't kind of handle the newer stuff. But 2.2.2 is still fairly recent. I think they're up to 2.4 or something. And then we load in these other software tools as well that are required by AlphaFold. All right, then the next part is we change directories to the folder where we want to run our analysis. And this is generally where, in our case, actually, yeah, so this can be anywhere. You could navigate to Scratch, you could navigate to your project folder. So that's just an example of where you might run this. But this shortcut, which becomes available when you submit a PBS job called PBS_O_workdir. Oops, I just copied something. Control X, undo. How do I undo? Cut. Sorry, didn't need to do that. So this shortcut it basically says whatever folder you submit your job in, navigate to that directory. So that's a little shortcut for you. All right, and then in the script also, I just make a backup of this script name in case you make any modifications to it. It automatically gets saved. So you can remember, oh yeah, these are the configuration parameters that I used in that particular script. So it's good practice to kind of back that up. I generate an output directory, and then we specify where the databases that AlphaFold requires are on Artemis. And in this script on here, we actually generate our own FASTA file. So you can replace your sequence there, and you put in multiple sequences as well. So I think separated by a header. So a header and then a sequence, and a header, then a sequence, I think is how to put multiple in. And then finally you get to the run AlphaFold step. So here is the run AlphaFold step. Then there may be other processing steps that you do along the way, especially about like maybe picking the right FASTA files and modifying them. Okay, so then we submit this job to the queue by typing qsub alpha_job.pbs, and that submits it to the queue. And then it will go, if I type qstat, I should see, yeah, my job is queued. There's another 3 running. And so if you are on the big Artemis, that will print out sort of hundreds of jobs, because there are hundreds of jobs at any one time. So you can just say and specify your own unikey, and that will give you sort of more detailed information about it for qstat. Okay, so when that finishes, let's see. So when that finishes,
Nate?
Yep.
Hi, how do I kill a job? Because by mistake, I had it at 20 hours. I've created a new job with shorter time, but if I wanted to kill the previous job.
Thanks, Lana. Yeah, qdel. So qdel, and then if you have the job number, so that one's my job, I can say 466194. I can,
okay.
oops,
Yeah.
I can go qdel 466194, and then it will kill that job.
Okay. Done. Thank you.
All good. Okay, so sorry, and I missed okay, so I've covered wall time. Katie's question in the chat is the only difference between this job and the little demo we did on AlphaFold, the length of the protein. Seems to be a big difference between that job. Yes, absolutely. So there's a big difference. So AlphaFold uses a different mechanism for searching, I think, databases. So, and ColabFold kind of skips that step. And so I think the main thing is that it's not as accurate, but what you lose in accuracy, you make up for in time. So depending on your requirements, I think the ColabFold is pretty good, and it's so much faster than AlphaFold in my very limited experience with this. So yeah, the ColabFold, and we can run ColabFold on the command line as well, which we might have time for instead of running the full AlphaFold. Okay, so let's all right, so we've run a job, you submit it to the queue. Okay, then some output is generated. So the output that gets generated, I have here this job01 afcpu.e and then my job number. This one .o and then the job number, and this one .o and then the job number followed by usage. So these 3 files are basically they're Artemis specific. Most supercomputers, they will generate sort of these diagnostic files. You can change this so it doesn't appear like this, but the default behavior is that it's the name of the job followed by EO_usage and the job ID. All right, now in each of these files, they're just text files. So we can have a look at those in something like Nano. So I'll look at this error file to begin with. I can see there's it's printing stuff, even though it's called the error file. I think most of the AlphaFold output gets put in this file. So this often contains like warnings and just things that would normally get printed to the screen, but they end up in this file. And you can see, yeah, some of these are actually errors. They, I think I have to scroll across. So, how do let's go. Yeah, so it's saying using pre-computed tools, saying it can't find a TPU, can't find a GPU, which is expected because I don't have a GPU set. And it's just giving me some diagnostics about the run. Finally, it says it's starting the jackhammer phase. So this is one of the tools it uses, I think, to search the UniProt database, and that gets started. But then after 10 minutes, I only set my job for 10 minutes, it gets killed. So I get this error at the end saying PBS job killed because your wall time was exceeded. So it's important to kind of look at these files to help understand, you know, what went right and what went wrong. So that's the error file. There's also the output file. Now, AlphaFold doesn't produce much, as I said, output in the output file. For some reason, it writes to standard error instead of standard out. I don't know why that is, but most other software kind of splits the two sort of evenly. So if you get warnings and errors, it'll put it in the error file. If you just get normal diagnostics like, hello, you've reached this part in your script, it'll put it to the output file. But anyway, between those two files, you can normally tell everything that would usually get printed to the screen ends up in those files. The last one is a usage file, and this tells you kind of details just about your run on Artemis. So it says, you know, how many CPUs you requested, how many GPUs, how much memory, how much, and then it says how many CPUs you actually used and the memory that you actually used. So you can use this to kind of figure out where you can improve efficiencies and where you might need to request more memory. So I've found the first phase of AlphaFold, it's very CPU intensive. The second phase is heaps memory intensive, so 32 gigabytes isn't enough. You kind of need sort of hundreds of gigabytes for it to run fast, and it runs a lot faster with the more memory that it has. And then only the final phase uses a GPU, so it can be pretty inefficient to use the GPU on AlphaFold directly. Okay, so and then there's a few other metrics in there that you can read it's just diagnostics about your run. Any questions about those? So that's general Artemis output. That's the same for any kind of job that you run. But then we have the actual output that gets generated from AlphaFold. So by what is set up in that script, it will produce the name of the job underscore output. You can modify that to anything you like. And then it will produce various output in there. Now, because my job was killed very soon, there's probably not a lot in there. Actually, the name of the FastA file is input, so it breaks it up by FastA file, and then it has some information about the MSAS step, which is currently empty because it didn't have time to generate any output. But here's one I prepared earlier. So example output, I have an AlphaFold example output, and when that job actually completes, you get these files. Now, the key ones, I guess, are probably the PDB files, and this is the predicted structure. So we can have a look at this. Let's see if this works. If I do module load, what is it, PyMol? You can use a bunch of different viewers, whatever you like. 2.4 PyMol / 2.4.0. So I'm loading PyMol, and then I think if I just type PyMol and then the name of one of these, it will should maybe display. Now, it might not work for you, because you need to do some fancy configuration, or not that fancy, but it's really different depending on each person's computer and everything. And it uses something called X forwarding. So, okay, so actually I haven't connected to Artemis using a X forwarding. So that means I can't view this directly. And that's because Artemis doesn't have a screen. It's like plugged. It has no screen. So you have to connect with that -X option in order to say, rather than printing to the Artemis screen, which doesn't exist, I want you to forward all the output to your local machine and display it on a screen there. So yeah, that didn't work for me. It may work for you if you've connected and you have an X server. I can demonstrate that. Maybe I should. So let me make a new connection. Just open up another terminal. Oh yeah, I'll open up a new one. So SSH -X -X. So the X that we put in here that says give us an X server, but you also need an X server installed on your local machine. So that's why it might be tricky. What the ICT train one, hpc.sydney.edu.au, and sad green caps 678. Okay, and what directory was I in? This one. So let's go to, I'll just navigate to that directory and see if this works. I need to type CD. I didn't type CD. I'm going to module load PyMol. Like I said, there's lots of different PDB viewers, but if you just want to have a quick look whilst you're on Artemis, I think this one is pretty reasonable. Otherwise, you can download the data later. You can move it over to RDS and look at it locally. Okay, so I've loaded it, and now I can do PyMol relaxed model 1. Sure, let's do that. All right, and so the output there, yeah, the PDB files are the predicted structures using different. Oh, I got an error. I've got lots of errors. Okay. Okay, I'm getting lots of errors. Maybe this will load. It's going to load. Oh no, it did. Yeah. So generally, we're connected to what's called a login node, but you also it doesn't have much computational power behind it. So sometimes you need to submit like a little mini interactive job in order to do even just things like loading up a viewer like this. Is it going to load? Might load. Well, that's going. Yeah, I'm not sure whether it's actually going to work or not. Hopefully, should show the full thing. Can I go file, open? Give me one of these. Okay, and the other things that are there are in that directory. Whilst I'm unable to load features.pickle. Oh, I must have. Oh, okay, so it's missing some of the data that it needs under it. Unfortunately, what have I missed? I may have messed something up. I'll see if I can find an example. Okay. All right. And oh, sorry. There's one more directory called in the MSAS directory, and this has the output for all those little sub programs that AlphaFold uses, and all of these are basically I can say head. These are all just various text files. Oh, I think. Oh, sorry. Head. Sorry, CD MSAS. Make sure I'm in the right directory, and then I can just look at the start of that, and so you can get more diagnostics out of those as well, and understand what's going on in these other files. Okay. CD. Okay, so yeah, I'll see if I can come back to looking at that, but maybe other people can have success with it. Oh, you had a look at it. That's good. It did work. X server on Windows. So yeah, on Mac, this is explained in the yeah, how to connect with the X server is explained in the setup instructions. But just kind of as a throwaway, there's the optional recommend installing Xports, and Windows has VCXserve. In my experience, is the most simplest. So yeah, hopefully that works for you. Okay, any other okay, how to download it. Actually, that is missing from the notes, so that's a good question. Let's get to that. Okay, yeah, I'll explain how to download these files in a second. What else is there? So there's the description of all the output files, and you can have a look at the documentation as well to see what everything does. And there's another example down here with the GPUs. And so there's very little to change if using a GPU as well. So once you learn, what part of your workflow is taking the most amount of time, you might consider adding a GPU. Keep in mind, yeah, there's far more CPUs on Artemis than GPUs, and far more RAM. So GPUs are kind of in high demand, and they're even if you request one, you'll probably have to wait a little bit, whereas CPUs and memory, you can request quite a bit and your job will probably start straight away. But yeah, the only difference is that you change this to one, and there's a couple of extra lines. So you have to just install these 2 bonus packages, and then set, oh, that's actually a mistake. You have to set the GPU relax to true. So it will perform that relaxation phase on the GPU, which is substantially faster. Okay, for that particular part. Oh, that's awesome, Lagana. Thanks for posting that in the chat. Yeah, that's okay. So copying it to our local machines. There's quite a few ways to do it. I think I don't have this in the notes, but one way is to copy it to the RDS and then look at it how you might look at things on the RDS. That's probably a recommended way. Another way is to use a tool called FileZilla. And this is good because it works on any platform, and so Windows, Mac, Linux, and it's fairly lightweight. So I think I already have it on my computer, FileZilla, or maybe not. Oh, okay, I'll just install it then, because like I said, it's lightweight. And I had it. It's okay. And this is a handy way. It's what's called an SFTP client. So we connected with an SSH client to Artemis, and an SFTP client uses file transfer protocol to connect to Artemis, and then from there we can Zilla. Okay, cool. So let me bring that over to the screen so people can see it. So this is FileZilla. And now if I want to connect to Artemis, I can go hpc.sydney. Oh, sorry, I need to put this in sftp hpc.sydney.edu.au. The username ICT_HPC train1. The password sad green caps 678. And I'll link the port. It should default to 22, I think. And it's saying, do you want to save my passwords? Sure, you can save it or not. All right, so this is Artemis on the right-hand side and my local machine on the left-hand side. And so now I can go to /project/training what do I call it? AFNAT. And then I can just copy that output over to my local machine. So if I just go, so you can just put that wherever, and there we go. I now have it on my local machine, so I can open up let's see, what was it called? Still going. AF demo, I think it's called AF demo. There's my AF demo. And I can have a look at the output. Although, yeah, I don't think I have a PDB view on my local machine. Oh, yes, I do. There we go. Oh, that was typical. Let's have a look. Maybe I do. PyMol. Oh, I have PyMol on here. Fantastic. So yeah, that's one way to do it. I'll post a link to oh, I haven't activated it. There we go. So I'll post a link to the instructions on how to do that as well. Okay, let's go, which is in the Artemis HPC training, the research data store, and it's this one, transfer from local to Artemis HPC. That's well documented there. Okay, so we only have about 20 minutes or so left, but you can see how to submit a job on Artemis, how to look at the output. Are there any other sort of high level questions? You know, having a go at this, how do you input multiple sequence in the FASTA file? That's a good question. So probably the best answer is on here. Let's have a look. FASTA. Multiple provide input with multiple sequences. Oh, so you need to include the model preset to be Multimer. That makes sense? Yeah, so if you have multiple sequences, then you need to also adjust another option on in the list of options there. And yeah, as Jake said, there's a bit of trial and error and sort of understanding all these various options. I think they're not all listed here. Some of them are actually in like the source code. So you have to kind of go into this run alphafold.py, and then in here, this is all the different options that you have. So there's quite a lot listed there, but at least it explains, how they work. Or that's the only place that I've found where they're nicely documented or documented at all. So yeah, that's in the run alphafold.py itself that we're executing. So the instructions on the AlphaFold page, they actually explain how to run this, and they recommend a tool called Docker. So Docker is like a container technology and a container is something that lets you, there's a few things it does. It's designed to have like a self-contained workflow. So you start from like a base operating system, and then in there you have the different tools and versions of tools installed, and then finally you have your final thing, which in our case might be AlphaFold. And they're all kind of tied up together. And so you can take that anywhere and run it on any platform and it will work exactly the same. So it's really handy. People use it a lot in sort of production for like deploying websites where you have, I don't know, complicated stacks of things and you don't want to have to deal with, multiple applications running on your website, all requiring different versions. So you just put it in a container and it works like that. I think that's what it was kind of designed for. But the research community goes like, hey, this is great for reproducing, making reproducible workflows, because anyone should be able to run this anywhere that Docker can be run. The problem is Docker has to be run as a root user. So you can't run Docker on Artemis. Instead, we use something called Singularity. And so Singularity kind of takes a lot of what Docker can do, but kind of restricts some of the, I guess, the power of it, which is okay. It's still very cool. And so the next section in the notes actually talks about this. And this is how we run ColabFold on Artemis as well. So basically, we've already made a ColabFold notebook. And so all you have to do, you can either download we've already made a ColabFold container, And so you can actually download that container directly onto Artemis and then just run it run it directly, or you can go and configure it and rebuild it yourself if you want as well. So there's the option there to rebuild it. But yeah, so I'll talk a little bit more about containers. So if I actually go to, let's go to this one. So if I go to Docker Hub, and let's search for the Sydney Informatics Hub, AlphaFold. There we go. So Sydney Informatics Hub, AlphaFold installation. We have there's some instructions there for how to actually run it. So this is all in the notes anyway, but this is for, I guess, anyone in the public, and it explains, you know, each of this particular version of ColabFold. It's built on like a really old operating system, and that's because Artemis kind of, because it's so old, it needs like special configuration, which is super annoying. But the idea is that if you can do it on Artemis, you can kind of do it anywhere else, because it's, yeah, all newer things should work in other places, but the really old stuff should work here. And there's a few different versions. So I think there's ColabFold 1, 1.3, and also AlphaFold 2.2.3. So the full AlphaFold with a slightly updated version. So I wouldn't recommend using that because we kind of have 2.2.2 on Artemis already natively. But the ColabFold one, and I should point out the versions don't match up with the versions on Artemis, so that doesn't matter too much. Sorry, the versions of ColabFold don't match up with the versions of AlphaFold. So 1.3 is not the same as AlphaFold 1.3. Okay, but I'm digressing a little bit. So the point is, these are stored here, so you can go and play with these if you like, or you can, do I have the source code is also listed there, so you can see kind of like how it's constructed. You don't really need to know that, again, if you just want to use it, but it has the instructions, like, you know, we install, we start with this Ubuntu 16.4 image. We then install Python. We then install all those, we install CUDA for GPU stuff. We install some PDB things. We install that K-align and HH suite that we used on Artemis. And then finally, we install ColabFold. And then there's some other configuration steps. But they are all there. And so that's really good for reproducibility. But then to actually run it on Artemis, if I go back to Artemis and I have this alpha_sing.job. So that is the Singularity version of AlphaFold, or in this case, it's actually ColabFold. So you can set up your arguments just like you did earlier. All you have to do is load in Singularity and also this thing called CUDA, which is the GPU driver stuff. We change directories, we make a new directory, we back up our script, we generate a faster file, or you can read it in directly from disk. Okay, and then the fun part here is actually the execution. So maybe I'll go back to, it's written here. So the execution, it just starts a little bit differently in that you say, I want to run Singularity. So I use Singularity run. Is ColabFold compatible with multiple sequences? I believe, yeah, I haven't tested this version. It should be though. So it just requires the right command line inputs, I think. And yeah, so basically you point to the image that you have to download or build. And then finally, after that, you submit this ColabFold command. And this is much like the AlphaFold command that we had. It takes much fewer inputs. I think, yeah, we have the FASTA file input, the output file, and then You don't need to point to any databases or anything, but you can set these other options, just like we saw on Google Colab, where you can set whether it uses templates, the number of recycles. So yeah, as Jake was saying, 3 might be good. 1's probably not enough, but I've just got 1 there for a demo. Do 3, 25, and so on. And all those other options that are available on the Google Colab, they would also be available in here. So, all right, and that is all documented. If you want to use that, that is on the Colab site. Now, I think if I try and submit this, oh yeah, it did work. It'll submit, but it'll come back with saying like you do not have access to Singularity. So if that's something you want to do, you have to actually get access to Singularity. So you can put in a request. I think I have. It was in the setup instructions on how to get a request to Singularity. So please log a request. However, that is, one way to run ColabFold on Artemis, but let's just talk about Guardi for a second. So Guardi is the national HPC, which is 10 times larger than Artemis. It has a lot more kind of to offer about it. And it's really well documented in the Artemis documentation. So just link here so we know where we're up to. Sorry, in the Guardi documentation is well documented for ColabFold, and it gives you a few examples. So they're running version 1.4 on here, and it says how these different, you can use ColabFold search. However, they say don't use the GPU on this one because that one doesn't use GPUs, but use multiple CPUs. And then the next step, the actual protein prediction step, that's where you should use the GPU to search for it. I don't know, to be honest, Lagana. I think always start with ColabFold, and then if you sort of need maybe more configuration options, then go back to AlphaFold. And it also depends where you're running things. So yeah, if you're comfortable, if you're using Guardi, like they only have ColabFold, they don't have AlphaFold. So it kind of depends. And I don't think Jake's not on here, so he I know like generally uses ColabFold more frequently than AlphaFold. And all right, okay, so they've got a few versions on there. And also the cool thing about Guardi is that you can take any Singularity containers there as well. So if you have a particular version or you have some other software that you want to package in, you can do that. Now, we've got only a couple minutes left. I thought I would just point out, let me close these as I go here. So that's Guardi. If you want access to that, you can ask me. Oh yeah, you also have to request access to that particular project, which contains ColabFold, which is pretty straightforward once you have access to Guardi. But if you need access to Guardi, you can kind of get a startup allocation. We run various merit-based schemes to get access to Guardi. So Sydney Uni buys a whole bunch of hours as a group, and then we allocate those internally. And there's also the big external ones. Actually, on Tuesday, the NC Mass, which is sort of the biggest application for the year, closes. So if you need lots of compute, make sure you get that in next week. Okay, so that's Guardi and ColabFold. Now, if you have money to burn and you want your own bespoke version of the latest cutting edge versions of AlphaFold, you do have the option to set up your own machine. So Sydney offers something called the Ronin Research Cloud, which basically gives you a virtual machine. Guardi is it's generally merit based, so you get access to various merit schemes, which generally are free. You can buy in hours from Sydney Uni as well. So through us, you can buy hours, and it's like $11 per KSU is the general rate, which is basically the cheapest thing you're going to get So if you're going to purchase hours anywhere, Guardi is very cheap. So that's good. But yeah, you can get a lot of free hours on it as well. AWS, on the other hand, is expensive, and but it's very good. So if you need like a solid machine that you can share between people, or you want to share between people, you have a specific use case, then having a dedicated virtual machine set up can be really useful. It can be cheap as well if you depends how you use it. So if you like shut it down when you're not using it and turn it on when you actually want to use it, then this can be a really efficient way to use it as well. So that option exists. So talk to me about that. If you outgrow the Artemis installation, or you're fed up with the queue times or anything like that, then we can help set you up. And this is probably, it might be slightly more expensive than having your own machine in like a local lab, but it's infinitely more elastic in that you can keep adding hardware as you need it, keep adding users. The security is taken care of. So it's so much more simple to use than having a local machine. So that's kind of the benefits of the cloud. And yeah, we have affiliations with Ronin, so we kind of have it's cleared for not highly protected, but just protected data. Whereas public clouds are, they're a bit more rogue, but you can do it there too. The last thing I'll talk about is the Australian Biocommons and Galaxy Australia. So Jake mentioned this as well. This one is a free one, and it's accessible to use. And I'd actually recommend this for simple stuff if Colab doesn't satisfy your needs, because the limitations that Colab have, the Galaxy installation doesn't. And I think this actually runs on Guardi in the back end, so it can be super handy. However, the options that you have available, I'll just make my window bigger so I can see. So this is Galaxy Australia, and maybe Nandan can talk about this if you're still there. It has a whole bunch of tools that are designed for mostly like omics research. And if you need to use any of those tools, it's like a point and click adventure, which is fantastic. And you can go AlphaFold. You have to request access, and I think that is because they're using kind of external resources, and GPU resources are kind of expensive relatively, so they just keep a control on who has access to it. But once you have access to Galaxy, which is free for Australian researchers, and it just takes a couple of clicks, then you can fill in another form and get access to AlphaFold, and you search for it, and then you put in you can upload stuff. So this one, yeah, it's very, it is limited. Does it have, it has some of the options available, but I think, is there anything else? Yeah, there's not really that option. So you have the templates, the sequence, whether you want a full database search or reduced one, and then the model preset. So that's basically it. I don't think there's any other options for that one. So that's why it's it's restrictive, I suppose. But you can sort of submit many jobs. And do we have one handy? Let me have a look. Oops. I was gonna just put one in. How about we put in our sequence from before? Paste sequence. That work? That's optional, reduced, and you click run. And that's it. So then it gets added to the queue, and then you get an email later when it's finished. And it's very cool that you can actually you can then share your results publicly, and they have really nice online viewers. So that was one that I ran earlier, which is kind of neat. Where can you find the information on where to tweak argument? So yeah, that's tricky. So either talk to Jake. He's my source of knowledge for this kind of thing. Read some papers, read the documentation and the source code. They're probably your best options for what the various things do. So it is all written in here, you know, it's dense, but you know, here we go, the model of, sorry, there's some of the parameters, but where you go and pick the options, it might explain the relaxation step can help be run on a GPU. This can be controlled with that. And so, yeah, some of those options are described here. I think that's probably your best bet. That's the one. So I think, yeah, we have many links in the training material. Oops, what was that one? So we've got lots of links scattered around the training material. Oh, whoops, that's not what I want either. AlphaFold, this one. There we go. And so, yeah, browse through that to have a look. Sorry, was there a question or something? Oh, okay, so Git, there's AlphaFold. Let me get that one. So there's the AlphaFold wiki. I was very confused by this as well when I first started this journey. There's the AlphaFold, which is the original AlphaFold. They're the developers behind most of the stuff. And then there's ColabFold, and their GitHub is the one that produces that the Google Colab that you saw, and I think they explain maybe what it is. Maybe they don't explain what it is, but basically that they've taken the AlphaFold source code and they've just modified it slightly to make it streamlined a little bit, remove some of the extras and simplified it. But they're still very much based on the same principles. Yeah. Oh, so yeah, if we love being acknowledged from the Sydney Informatics Hub. You can probably cite some of Jake's work if you can, but yeah, there's nothing in particular. I think just if you use AlphaFold, the only thing that's required to be cited is that original AlphaFold paper. I don't even know if ColabFold requests citations, but yeah, you should do those. But yeah, if you enjoyed this training, be sure to acknowledge us. We always appreciate that. Lets us produce more things. Okay, so I think that's basically everything. I'll stop talking in a minute. If everyone could, there is just if everyone could fill out a survey, we'll send out a post-workshop survey at the end of things and get everyone's feedback. But if you have other questions, please feel free to send us an email or anything like that, and we'll see how we can sort of help you from there. Nan, did you want to say something?
Oh no, sorry. My puppy came around, sorry. And I was unmuted.
All good. Yeah, we've got a team of bioinformaticians and networks to other people who are using AlphaFold across the university as well. So please get in touch if you have any follow-ups. Good luck, everyone. We'll post the recordings probably next week, I think. All right. Thank you all.